or the most current idea about viruses, especially the ones like viroids, while they are not on the tree of life, they may have given rise to the tree of life. Here is something that is a free-floating RNA, and there's always been that question: could just RNAs form without a plasma membrane around them, without ribosomes around them? And obviously they can, because here we have examples of them. Examples of free-floating RNAs that cause potato disease, that cause grape disease. Well, the idea is then that could be the progenitor for life, and then that got encapsulated with fats, and we start having some processes that led to metabolism and all those wonderful things that we define as life. So, if the question came up about viruses in the tree of life, you'd say, well, they're not on the tree of life, but it's possible that they gave rise to the tree of life. So that's something to consider with our viruses. Another thing that we see that differs with the viruses are how they are able to enter host cells. That is really an interesting thing about viruses, is they are very species specific, and it is very uncommon for a virus that makes your dog sick to make you sick. If your dog has the sniffles and is sneezing, or if your puppy gets parvo, you're not going to get parvo. You're not going to get a cold from your dog. The most common viruses we deal with don't cross species readily because they are so specific for getting into a given host cells. There are some notable exceptions. Rabies is one of the best examples of something that readily crosses species. And we do know that with flus, we are concerned about the viruses getting the ability to jump species. Some of them can, some of them can't. The ones that are able to especially jump from avians into us are really concerning, because the viruses tend to be housed, or the flu viruses tend to be housed in avies, tend to impact the lungs much more significantly. And so there's been, for the last few years, a big uproar about bird flu. Oh, when's the bird flu coming? And why do they care? The swine flu didn't do that bad of a job. I mean, it was scary, but didn't kill any more people than a normal flu year. So why are we so worked up about avian flus? Well, avian flus tend to target the lungs, and they tend to hit the lungs hard and fast, and they tend to make the victim end up drowning in their own fluids. And so it's not a pretty way to go, and there's not a really effective treatment for drowning in your own dying lungs. So that's problematic, and we're really taking a stroke at that right now. So what about the information flow? The information flow, the basic one we see at the bottom, DNA, RNA, protein, which is really just very stripped down. By the end of the semester, hopefully you'll appreciate that is a very, very stripped down information flow. But that's the same in all living things. The viruses do some wacky things. We'll talk about that. But living things, they have a blueprint of DNA. That DNA is transcribed into a message that can be interpreted by our ribosome. That's RNA. That message can be interpreted by the ribosome, and the ribosome can take that message and build a house front, build the protein. Remember, golden answer is always protein, because if there's something that is being done, if it's an activity, 9 times out of 10, it's going to be protein. Now, within that information flow, there are some nuanced differences between the prokaryotes and the eukaryotes. Our prokaryotes, we see the DNA usually has a lot less bells and whistles attached to it. It's called naked DNA. I like to think of DNA during the summer in Miami. It's just not wearing a lot of clothes, but to stay legal, it's got to have a few. So think about the DNA in the prokaryotes being kind of a stripped down version. The DNA is still the DNA. What we're talking about here is the clothing that helps package it to get it to fit into that space. So it still needs some degree of packaging. Now I've got something packaging here that ends with an ing, so it's a doing kind of thing. What do you think involved with that packaging process? If the DNA is being packaged, what is the packaging? So hopefully you're thinking golden answer right now, and that would be proteins. So our prokaryotic DNA doesn't have a lot of proteins associated with it, as opposed to the eukaryotes, which are like, you know, it's like Michigan during January, or apparently it's like Oklahoma last week. You want to have lots of layers and clothes and lots of proteins associated with that DNA to make sure that everything is packaged into that nucleus. A couple of other key differences. In our prokaryotes, we usually only have a haploid chromosome. That means one copy of a chromosome, typically only one. It's typically a circular chromosome. Now, there are some exceptions as far as the number of chromosomes present. Vibrio cholera, hopefully you remember that from the quizzes. The Vibrio causes the disease cholera, and the scientific name is the Vibrio cholera. That organism actually has 2 chromosomes. Now, it's still not diploid because there are 2 distinct chromosomes, chromosome 1, chromosome 2. It does have more than one, and it is kind of a rare case. Typically, eukaryotes are diploid, and now that means you have 2 copies of each chromosome. Like I have a copy of chromosome 1 from my mom and another copy of chromosome 1 that I got from my dad, and so on and so forth. Also, your eukaryotes tend to have a linear chromosome. That means it's kind of from end to end is separate, so that you have this line of DNA. Your circular prokaryotic chromosome